This morning I want to talk to you about the future, but it's a future that's not very far away. I want to talk to you about new research in the laboratory that aims to eradicate genetic disease. Now, to give you a bit of background to this, I want to let you know that genes and the DNA in our bodies, and in fact in all life, shapes that life. So whether that's bacteria becoming resistant to antibiotics, or when you go down to Point Isabel and you go to the dog park and you look around at all of those dogs, they're all dogs, but they all look so very different from each other, the same way people look so very different from each other. That's all encoded by genes. And if you look left and right in the audience, this is maybe going to be a little awkward, but look at the people's ears, okay? You'll notice that people have attached earlobes and detached earlobes, and that's also coded for by genes. Now, the same way that genes build us, our bodies, earlobes, dogs, they also, when they go awry, can lead to problems. This is David Vetter, who you may have also heard by the name the Bubble Boy. He was born with the mutation in his DNA that led to a disease called severe combined immunodeficiency, aka SCID. Now, the result of severe combined immunodeficiency is that you're born without any immune system whatsoever. So you're incredibly susceptible to infection. And you have to live in an isolated environment so that you can't be exposed to anything that might cause an infection. Now, it turns out there are many, many genes that can cause this single disease, and we're still learning a lot about them. Another genetic disease, but one caused by a single mutation, is called sickle cell disease. Now, this one is particularly frustrating. This is the so-called first molecularly known disease. We have known the mutation, we have known the cause of this disease since 1949, okay? This single mutation in a protein called hemoglobin causes red blood cells to curve up, and they get stuck in capillaries and cause them to clog. And this can cause severe pain, it can cause organ failure, and if it happens in the brain, it can cause strokes, okay? So we've known about this forever, but we haven't been able to do anything about the underlying cause. And that's because whenever you take a drug, when it comes to genetic disease, you're really treating the symptoms. The problem lies in your very DNA, but the drugs are just blocking the symptoms that lead to the disease. And so if you stop taking the drug, then the mutation is still there, and the symptoms will come back. So, what's happening now, and really getting biologists very excited, is the ability to go in and try to reverse the very mutation itself, to try to remove the fundamental underlying cause of genetic disease, and thereby not just treat symptoms, but to truly cure genetic disease. Now, the real problem here is knowing how to make the changes. If you think about all the DNA in your body as a big library of A's and T's and G's and C's, we've had the technology for the last couple of years to read that very quickly. So through advances in something called next-generation sequencing, we can read through this library very quickly. But if you imagine somewhere in this giant library, there's a single book with a single typo in it. We can find that now, but how do we go in and how do we correct it? And new advances in gene editing are promising to put find and replace right on top of there. So now, not only can we find this thing, we just say, oh, now we found it, we need to edit it, okay? Now, I want to be really clear about this. So in the news, you may have heard of a couple of different types of gene editing. And there's been a lot of discussion recently about something called germline gene editing. This is something where you edit a gene and it gets passed down onto other generations. And that is not what I'm talking about today. Now, I want to make it really clear about this because actually germline editing is quite difficult. It's much easier to do non-heritable gene editing. And in fact, the National Institutes of Health, which funds most medical research, will not support germline editing. So nothing that I'm talking about today is this quandary of do we make edits that are passed down. Instead, I'm talking about somatic gene editing. This is just like giving someone a drug. It stays in that person, but it's a cure for their disease and is not passed down to their children. And when we talk about gene editing, what we're really talking about is gene cutting. We need to find the bit of DNA that leads to a genetic disease, cut it out, and replace it with the right sequence. And so how do we do that? The way we actually go about doing that is using this. So this is the thing that has everybody all excited. This is called Cas9, and it's part of something called the CRISPR system. This was discovered as part of actually a bacterial system, comes from bacteria, and has been repurposed for gene editing. In this white part is the protein, aka Cas9. The blue part is the gene that it's targeting, the DNA. And if we pop it open a little bit, inside is something called the guide RNA. And this is the bit that makes this such an important technology. Gene editing has been around actually for decades, but it's been very hard to make it target different genes. And so the ability to say, oh, I want to target this person's mutation, or this person's mutation, or this person's mutation, it's been very hard to change. Whereas this guide RNA recognizes the target gene with simple rules of base pairing, which you probably learned in high school. A goes with T, G goes with C, that kind of thing. And so if you know A goes with T, G goes with C, you could potentially design one of these things if you knew the sequence that you wanted to target. Once it's been designed and goes to the right place, Cas9 then will make cuts in the target gene, and you can get replacement of that sequence. Now, where should we edit? Right? Where should we make these edits? And I'm going to tell you about 2 projects that are going on in my lab in collaboration with other labs to try to understand more about genetic diseases and to try to cure them. First off, if we think about what type of tissue to make edits in, we want to be having an effect in 2 different types of cells. For sickle cell disease, we need to have an effect in so-called erythrocytes, or red blood cells. These are the things that curve up and cause the clogging. And severe combined immunodeficiency, we need to be doing something to B cells, T cells, and natural killer cells. These are the business end of our immune system. But if you look all the way at the top of this, you'll see things called HSCs, or hematopoietic stem cells. Now, you heard about these earlier this morning. Hematopoietic stem cells reside in your bone marrow, and they give rise to all of the different types of cells in your blood. Okay? So these little engines that turn into all of the different types of cells that do what your blood needs to do. And so to cure sickle cell, to learn more about SCID, we're going to make edits in the HSCs. Now, there are ways to combat some of these diseases, for example, sickle cell, using bone marrow transplantation. You can do the same thing for SCID. Okay? This is so-called allogeneic bone marrow transplantation. You take bone marrow from someone who's unaffected, and you give it to someone with the genetic disease. And this ends up curing their disease. But there are some pretty severe side effects. You have to get rid of the person's original bone marrow through ablative chemotherapy. You have to suppress their immune system so that they don't try to attack the donor bone marrow. And there's also a chance of the donor bone marrow attacking themselves. And this is in addition to problems of finding the right donor. So you have to find matched donors, and this can be very, very difficult for certain diseases. And what we're hoping for some of using gene editing is that we can actually turn people into their own donors, to give them the power to be their own donors. The idea here is we're going to do something called autologous bone marrow transplant. We're going to take someone with the disease, we're going to take their bone marrow out, and use gene editing to remove the mutation, okay, to turn it back to a wild-type sequence, which is then going to be reimplanted back into them to cure the disease. Now, we're doing this together with really fantastic clinicians from UCSF and Children's Hospital Oakland. Here on the left, you can see Jennifer Puck and Alex Marson working with a child with SCID. And on the right-hand side, you can see Mark Walters and David Martin from Children's Hospital Oakland, with whom we're working on sickle cell disease cures. First, I'll tell you a little bit about what we're doing with SCID. Remember, we're working in these B cells, T cells, natural killer cells, but I told you there are a huge number of mutations that can lead to this disease. And at this point, as we sequence more and more patients, we're finding more and more mutations. We don't know how those lead to disease, how they lead to different presentations of the disease, and how we could possibly cure that. And so if these are the mutations we already know about, what we're starting to do using gene editing is to take patient-observed mutations and try to use gene editing to figure out how those mutations lead to those diseases, with the eventual hope of coming up with a way to attack those diseases. And that brings me to what we're trying to do in sickle cell. So with sickle cell, I told you there's a single mutation that leads to this disease. We've known for 50+ years exactly what we need to do, but we've lacked the ability to do it. And so in the lab, we have managed to finally create a cure for sickle cell disease. Now, I said in the beginning, this is a future thing, right? So this is in the lab, we're not in the clinic, and there's still a lot of work that remains to be done. But we're very excited about it. So we take bone marrow from patients. We use gene editing using Cas9 to change that mutation back to normal, and we put that into mice. And the reason we put it into mice is we need to know that these are actually stem cells. We need to know that they can go on and make hemoglobin proteins, they can make other blood types, things like that. And then, after a long time, we take that material back out of the mice, and we ask, is it non-diseased tissue? Is it normal human tissue? And the answer is actually yes. So when we take these stem cells back out after they've been edited, they make normal hemoglobin, and they seem to make normal blood cells. So we're very excited about these results, as you might expect, and our next steps are trying to find ways that instead of going into the mice, how can we get to going back into patients? So there's a long road ahead of us, but we're very excited. So what I want to leave you with is the idea that gene editing could totally change our relationship with genetic disease. Instead of genetic disease being something that you're born with and then you live with throughout your entire life and you just treat the symptoms constantly, we could maybe have a way to go in and cure those diseases completely. So I want to leave you with a couple of difficult questions. I think the first easiest one is, if you had a neighbor who had a genetic disease and they cured that with gene editing, how would you think about them? Would you think about them any differently? You know, they had sickle cell disease one day, the next day they didn't. Would you think about them any differently? What if instead of curing disease, they instead got an enhancement? So this is a cow which has a mutation in a gene called myostatin. If you mutate this gene, the result is incredible muscle buildup. This also happens in dogs. You may have heard of whippets and bully breeds getting this. And it turns out people can also be born with this. Okay? Now, this is actually a disease when you have this mutation because it leads to cardiac problems and other side effects. But it is possible that you could think about enhancements, maybe for muscle growth, possibly for sleep extension, things like that. These are much, much more difficult to do than diseases, which tend to be a single change, but it could be possible. Before you dismiss this out of hand, think way back to the beginning when we heard about people going to the stars. And remember about possibly sending people into space. One of the side effects of people being in space for a long time is bone loss, muscle wasting, things like that. And so thinking about one of these big futures, big ideas for the future, if we're going to send people to space, to live in space for a very, very long time, could it be that we need to be making these kinds of enhancements to people to allow them to live there? So I think this is something that we should all be thinking about. The future is very fast approaching. To me, the idea of curing genetic disease is a no-brainer. This is a chance to cure diseases at the source and to give people a chance at life without pain and to stop treating symptoms. The technologies don't go back into bottles. And a lot of people are already talking about what else we can do with this technology. And I really just want to point out to you in the audience that we as societies and we as groups get to decide what we do with the technology, right? We control our own technology. And so I think this is all up to us. It's not some train that we can't get off or some train that we can't steer. We get to decide where we're going to go. So the choice is truly ours. Thank you very much.